It is great to have Matthew MacEwan here today, who's an undergrad alum and has gone on a really interesting entrepreneurial journey. We were just chatting about some of the work that he had done at WashU and which helped lead to the birth of a company. All of our sessions are moderated by students, or in this case, recently graduated students. So Pravi, thank you for moderating today's session, and I'll turn it over to you in a second to introduce yourself and Matthew. For those of you that are joining a session for the first time, we love these to be as interactive as possible. So please let Pravi know if you have a question. If you're on Zoom, you can raise your Zoom hand or just put a note in the chat that you'd like to ask a question. If you're watching via LinkedIn Live, just put a note in the comment, and Julia and Doug and I will be monitoring LinkedIn and make sure that we bring it into the conversation. So thank you, Pravi, and thank you, Matthew, for joining today. And Pravi, over to you.
Thank you, Mr. Goldberg. All right, so today we have Dr. Matthew MacEwan. He is the Chief Security Officer of a company called Acera Surgical. And Acera Surgical is a company that specializes in soft tissue repair and wound treatment. And so now I will pass it on to Dr. MacEwan. And if you could just introduce a little bit about yourself, your position in the company, and what Acera Surgical's mission is.
Perfect. Thank you so much for the introduction, Pravi. It's great to be here, and thank you, Michael, as well. As Pravi mentioned, my name is Matthew MacEwan. I'm the Chief Science Officer and founder of Acera Surgical, which is a bioscience company based here in St. Louis, Missouri, which is developing a new class of nanofabricated and electrospun materials for soft tissue repair applications. As Pravi mentioned, I'm a CASE alum. I graduated with my undergrad degree in biomedical engineering in 2004 from CASE. Actually did a lot of work on the neuroprosthetics side back in the day with Dr. Grill. So it was great to hear about some of the folks who are currently involved in the BCI field. So always excited to hear what's going on there. But today I want to share about my story in finding my sort of application of biomedical engineering, and then taking this my skill set, I guess, that I developed at CASE, and taking it down an entrepreneurial pathway. So as I mentioned, I did my original studies at Case Western, focused in biomedical engineering, and specifically with a focus in polymer biomaterials. So I ended up working in the Kent Hale Smith building for quite a while. And got very interested in how you could use advanced tissues or advanced material platforms and polymeric materials for healthcare applications. And as I finished up my time at CASE, I was really sort of torn between taking those skills and applying them in the medical field, or sort of going down the research path, and was lucky enough to find a great joint degree program here at Washington University, an MD PhD program that I sort of got into right after CASE. So I came down here to St. Louis, went through the MD PhD program here at WashU. And while I was doing my PhD in biomedical engineering, I ended up working next door to a lab run by a gentleman by the name of Yunan Shah, who's now at Georgia Tech. And Dr. Shah was very interested in how we could utilize nanoscale materials to pattern and control cells in culture. We started looking at how we could leverage nanoscale materials to actually design better tissue interfaces or matrices that could be used to actually form and repair different tissues. And we eventually found this unique class of materials called nanofibers that were being produced using a technology called electrospinning, and found that these materials worked very well to mimic the structure and architecture of native tissue. And we started doing some just basic cell culture work, looking at how cells responded to these materials, and then how these materials could be used in repairing and healing of various types of wounds. And pretty soon after a few animal experiments, we realized these materials had some very interesting benefits and were working as well, if not better, than some of the existing tissue matrices that we were pulling off the shelf in the operating room here at the hospital. And so at that point, realized that maybe these synthetic and engineered materials really could offer sort of a new approach to how wounds are treated or tissues are repaired in the clinic. And so as a medical student, just decided that we needed to form a company around this technology as a bridge to get this technology into the clinic. And so I went to a few folks in the entrepreneurial program here at WashU, and eventually sort of found a small group of people, about 4 people, and raised a seed round of funding. And eventually we took this idea of using electrospun and nanofabricated matrices for tissue repair from the lab all the way to our first FDA approved product in about 2 years with about $2 million. And so from there now we've grown into a team of over 60 people and have 3 different FDA approved products and more on the way. And are really now making some great traction and bringing this type of technology out to the clinic. So very interesting sort of, I don't know, entrepreneurial path, one I didn't necessarily expect, but really great to see this technology now making a difference here in the clinics all over the US and abroad. So, so with that, now that I've sort of shared my life story, if you will, I want to sort of show you a little bit about the company, about the technology, and about some of the great work we're doing in the clinics here in the US. So with that, I'll show a few of my slides here and kind of give you a sense of the company that we've developed and what we're doing. So as I mentioned, so Acera Surgical is based still here in St. Louis. We were founded in 2013. And as I mentioned, are developing and commercializing these electrospun matrices for soft tissue repair applications. The exciting thing is this technology is all really backed by 10 years of academic research that we've done here at the university. And now we have a number of FDA approved products that are being used in the VA setting and the DoD setting, as well as in commercial clinical applications as well. Really, a lot of this work sort of started with our thought process of how could we use this technology to engineer synthetic extracellular matrix. So as many of you might know, the extracellular matrix is very important. It's sort of the structure and the architecture that cells live in human tissue. It's made up typically of ECM proteins that cells grab on and grab onto and grow into. But the problem is that in many cases, in many clinical applications, the extracellular matrix is disrupted. And this is particularly true in the case of chronic and difficult wounds, where enzymes, bacteria, et cetera, may break down the extracellular matrix. And when the extracellular matrix is gone, cells have nothing to kind of come and grow on and grow into to reform and repair damaged tissue. And so when you look at it, there's been a number of different ways that people have, or clinicians have tried to repair the ECM, ranging from autograft materials like split thickness skin grafts and rotational flaps, all the way to animal-based products and human allogenic products. But our thought process was, rather than harvest another tissue from another part of the body or from an animal, could we use sort of the latest in advanced material science to engineer a product that has sort of all the optimal properties that you would want in a sort of regenerative matrix that you could use to heal some of these soft tissue repair applications. And so to do that, we turned to this technology called electrospinning, which electrospinning has been around for a long time. And it is really uniquely able to make very small diameter fibers. And so we turned to this technology and really mainly because we found that this technology would allow us to create non-woven fibrillar matrices that look very much like and mimic essentially native ECM. So what we do is we take resorbable and biocompatible polymers, we dissolve them with solvents into a liquid, and then we shoot them out of these very small nozzles that you can see here on the left-hand side of the screen. And what happens is it pulls a very small bead of this polymer out of the nozzle. And then as that bead of polymer is stretched and drops over a small gap, it stretches these fibers to the point where they're nanometers in diameter. So very small diameter fibers, smaller than individual human cells. And then what happens, we just continually spray or deposit these fibers one layer on top of another until we get a three-dimensional matrix of these non-woven nanoscale or submicron scale fibers that you can see on the right. And what's really exciting about these materials is that they really mimic the same structure and architecture of native ECM. So even though they're synthetic in nature, they really provide the same type of architecture as you have in the human tissue. So here you can see sort of a zoomed in scanning electron micrograph on the left of our electrospun materials. And in the middle, you can see a similar SEM view of human ECM. So you can see that these materials have a unique ability to mimic native ECM in that they have the same sort of size scale, fiber diameter architecture of ECM. And what's also really interesting is when you look at some of the other xenogenic or processed human allogenic materials that are out there, a lot of times all the chemical processing that you do to those tissues actually changes their ultrastructure to the point where it really stops looking like native ECM. So in a lot of ways, our synthetic materials are able to better sort of mimic the structure of native human extracellular matrix. And that's really important because it allows cells to uniquely interact with these materials and essentially grow into them very quickly. This is the results of an early study that we did that shows just how quickly cells can grow on and grow into these materials. In this case, we seeded fluorescent fibroblasts around the outside of our material up in the top left. And then at time zero, allow these cells to grow into the material as quickly as possible. And within just 24 and 48 hours, these cells actually grew into the material and started to replicate on the material. So it really shows you how quickly cells can grow on and grow into these electrospun matrices. And that's very important when you're trying to bring cells into, say, a damaged tissue or into a wound and need those cells to come in order to set up new ECM and eventually new tissue. So this biological property that the material has, the ability to support cellular ingrowth and cellular retention, is very important. But another interesting aspect that we found was that the material is also just very easy to handle and easy to use. It's flexible, it's drapable, and it's strong enough to actually suture or staple into human tissue. And so this is a very important point because a lot of the surgeons and clinicians that we showed this material to actually really enjoyed the handling property of the material and found it was easier to use and deploy clinically than some of the other products that were out in the market. And so as a result, this handling aspect, this ease of use, really became another important point that we found people and clinicians really enjoyed. So with that, we started to look at sort of what were the potential applications for this material. We essentially created a whole list of different applications for this type of matrix and found a number of interesting ones that we wanted to explore. But we decided to start with one that was sort of close to my initial clinical work, which was in neurosurgery, and sort of focused our initial efforts to develop this technology as a means for performing dural repair. So first of all, the dura mater is the membrane around the brain and spinal cord, and during routine neurosurgical procedures, it's very common that the dura mater can be incised or ripped or removed. And in those cases, you need to put a matrix or a graft into the dura in order to repair and restore that watertight seal or membrane around the brain and spinal cord. And so initially, we thought that we should develop this technology for use in dural repair and sort of use this as a synthetic scaffold for performing these types of repair procedures. And so what we did is we took the material, we initially compared it to some of the existing dural repair materials that were on the market, including Duragen Plus, which is a bovine collagen product that's very commonly used. We found that our material offered sort of improved tensile strength and additionally sort of barrier qualities that that really were ideal for use in dural repair. And eventually we actually completed a large animal study comparing our material to some of the existing dural repair materials that were currently available on the market. And in this large animal study, we actually took our material and compared our Electrospun product to a variety of different bovine collagen products, including one called Duramatrix, which is one of the products sold by Stryker. So we left those implants in the animal, and we came back a number of weeks later to look at the degree of tissue infiltration and tissue repair that occurred following implantation of the material. And this is really a pretty interesting, study in that we found that, as shown here in sort of figures C and D, there were some significant differences between how tissues responded to our electrospun dural substitute material and existing cross-linked collagen dural substitute materials. What we found was, in this case, at 4 weeks postoperatively, we saw a lot of cellular infiltration into our electrospun matrices. We saw very little adhesions to the underlying cortex, very little inflammation compared to the Duramatrix material or the collagen material here on the right, which was so dense and so highly cross-linked that cells weren't even able to penetrate it. And so I think it really provides an interesting point in that how these electrospun matrices actually can promote and incorporate cells into the matrix very quickly and start to set up that new tissue that's needed to repair these types of, in this case, dural defects. So, so it was some really interesting animal data. We took that animal data to the FDA and eventually got FDA clearance for our first product, which we called the Cerafix Dural Substitute. This is a very exciting product, and it was really the first fully synthetic electrospun material utilized for neurosurgical applications and dural repair applications. And so this is, again, we now provide this to surgeons across the country. It's a soft, white, flexible material that they can suture into the dura mater. It sets up a matrix to form new dura, and then resorbs away completely over time, leaving just that newly formed tissue behind. So this is our first FDA-approved product that we developed using the technology. We now have used it at a number of different places around the country and internationally. I will say that the very first case was actually done at the Cleveland Clinic right down the street from Case Western. It was a very neat case in that it was actually a woman who had a pituitary tumor and which was sort of impinging on her optic nerves and was losing her vision. And the surgeon went in, removed the tumor, and then used our material to repair the dural defect. And that woman was able to get her vision restored, and the dura material did very well to prevent any type of complication post-operatively. So very interesting case there. We now have also used it at Hoag Hospital in some of these posterior fossa cases, as well as Harvard and Beth Israel in Boston, using these materials to repair the dura following tumor resection and even invasive sort of aneurysm correction. So we're seeing some really interesting applications for it. And it's just a great example of how a unique synthetic solution can be a real benefit for some of these surgeons who are doing complex surgical procedures. So with that, once we got our first product into the market, we started looking at other potential ways that we could utilize this technology. And the one that really sort of popped to the top of our list was in wound care. So it's not mentioned, I think, quite as often as it needs to be, but chronic wounds like diabetic foot ulcers, venous leg ulcers are a huge economic problem to our healthcare system, and additionally have very significant implications in terms of patient health and quality of life. And while there are a number of interesting solutions out there for treating these types of wounds, a lot of times even the most advanced therapies don't work in healing some of these very difficult wounds. And a lot of these wounds will lead to things like amputation or worse for these patients. And so our thought process was, could we use electrospun materials as a way to create new types of tissue matrices and graft materials that could help treat some of these very difficult and long-standing wounds. And so, so with that, we took our material, we re-engineered it, and we tested it out in a large animal wound healing model. So we created full thickness wounds in the back of pigs, essentially creating wounds that went all the way down to muscle fascia. We then treated those wounds with either our electrospun materials or some of the existing matrices that were sort of presently used in the wound healing setting, particularly in this case, Integra's bilayer wound matrix product. And the results were pretty interesting. So here you can see what the wounds look like after being treated with either the collagen matrices or our electrospun fiber matrix. We saw these wounds heal over the course of 30 days, but what's really interesting is that we saw that these wounds healed significantly more quickly using our electrospun material here shown in blue than using the existing collagen matrices shown in red. So that was one interesting finding, but even beyond that, we also found that the electrospun nanofiber materials we're able to heal those wounds with significantly better quality granulation tissue, more granulation tissue, and significantly less inflammation. So again, really interesting in that we're actually able with a synthetic matrix to get less of an inflammatory response than some of these existing animal-based collagen products. Also, another important point we found was that we found significantly increased neovascularization with our materials as compared to these existing products that we sort of pulled out of the OR and out of the market. So some really interesting benefits of the electrospun scaffold as compared to some of the existing sort of gold standards in the market. And this just shows you some of the results from the histological work that I think are really important, particularly here in the bottom. You can see in figure D, this is a view of the tissue or the wound site treated with our electrospun material at 30 days. And you can see that nice pink granular tissue, a lot of nice neovasculature growing into the wound, and a very low level of inflammation. And in comparison, on the left-hand side in figure C, you can see the ongoing inflammatory response against some of the existing collagen matrices that we compare the product to. So you can see a very different response and much better quality tissue with our electrospun matrices as compared to some of the existing technology that's out there today. So with this data, we went to the FDA and got our FDA approval for our second product, which we call the Restrata wound matrix. This is the first electrospun matrix approved for use in the treatment of a variety of different types of wounds. Again, it provides this unique nanofiber, non-woven structure that you can put right into a wound and allows cells to grow into that wound, set up new tissue, and eventually heal those wounds. And this has really become sort of one of our most important products. And we're seeing some amazing traction with this technology now in wound care centers across the country. This is a little bit about how we sort of use the product. So in a case like this, where you have a chronic diabetic foot ulcer on the bottom of the foot, what happens is that we take the material, we pull it right out of the package. It comes sterilized and can be stored right on the shelf. We cut the material down to size. You can fenestrate the material and put it right into the wound bed. You can then suture it or staple it or steri-strip it right in place. And then you can put on top of it whatever dressings you need. We typically recommend a non-adherent dressing and then dressings to sort of manage the moisture of the wound as well. And using this approach, we've seen some amazing healing and sort of therapeutic benefits for the material in a variety of different settings. So these are a few different cases that we, or a few different types of wounds that we've really treated with the material. We found it works very well in treating diabetic foot ulcers, like the one here at the top, venous leg ulcers, which are very difficult, typically very exudative, wet wounds, and even pressure ulcers, which happen to occur over the heel or the sacrum. So really, we're finding the material works in a variety of different wound care settings and works very effectively in treating a number of these very difficult wounds. From that point, we sort of branched out and started to do a number of different clinical trials now, looking at how how effective the material is in some of these settings. These are the results of two of the earlier clinical trials that we conducted. One was a retrospective study that we performed on the East Coast out with some folks in the New Jersey area. In that one, we treated 82 different chronic wounds, which on average were 36 weeks old. So these are very long-standing, difficult wounds that have about 78% of which had been treated previously with other advanced therapies. And using our Restrata matrix product, we were able to heal on average 85% of those wounds that had previously failed other advanced therapies. And we did so on average in about 5 weeks with about 4 applications of the material. And so that's a great result on par with some of the most advanced technologies that are currently out there. So it was great to see that initial result. We then replicated this study in a prospective multi-center trial that we conducted in the West and the Southwest, and saw 75% healing rates in chronic diabetic foot ulcers in that study. So again, just some great examples of how effective this this material and this product can be in some of these very difficult settings. And now we have a number of additional level 1 randomized clinical trials that we're conducting to further kind of expound on the efficacy of this material across a number of different clinical settings. So really excited to see those continue here through the end of the year. But with that, I just wanted to kind of show you all a few of the different wounds that we've treated over the years with this product, and just kind of talk a little bit about some of the positive impacts it's had on some of the patients. And so I'll just show you a few of these here. So first, these are a few examples of just diabetic foot ulcers that we've treated with the wounds. You can see sort of the progression here at the bottom. This is an 84-year-old patient with a diabetic foot ulcer on the heel. And you can see, I mean, this over the course of multiple weeks, this wound eventually went to full closure, even after it had been treated previously with advanced therapies. So it kind of just goes to show you that this material really works well, even in the instance where other products and technologies have failed. And that really is important for clinicians who need other tools in their toolbox to approach these very difficult wounds. This is another diabetic foot ulcer you can kind of see on the bottom or the plantar surface of the foot. Again, lots of sort of macerated tissue around it here to begin with. And you can see that wound gradually progress and close here. And eventually we were able to close this one right around about 8 weeks of treatment. Here's another interesting one. This is actually an ENT case where we had a large basal cell carcinoma removed off the side of a woman's face and treated that defect with the Restrata material. And we're able to get very good filling of that defect and closure of that defect with very minimal scarification. So some really interesting applications now for this material in that space, especially sort of the post-Mohs wound space, an area we're looking at quite a bit. This is another type of wound. We're seeing a lot of use for this also in extremities and removal of other types of tumors and cancers. In this case, we took the material, meshed the material, and put it right into the wound bed. And you can see over the course of multiple weeks, you can see the new tissue actually grow up and grow through the matrix. And eventually that new tissue starts to fill in the defect and eventually completely heal the wound. We're also finding that the material works very well in a number of advanced applications. This is a wound over the foot that actually has some exposed tendon. And so using the material, we've been able to heal some other difficult wounds like this. Also, this is actually another type of post-Mohs wound over the ear. This is actually there was a cancer in this gentleman's ear that was resected. We put a piece of our material on, and this wound eventually completely healed as well. Two more here just to quickly show you. This is a really interesting case. So this is a case from Texas, a gentleman who came in with a very advanced wound over sort of the first met head here on his foot. This wound was so advanced that the surgeons wanted to directly amputate his foot and potentially his leg because of how bad the wound was. But what happened was the clinician thought he'd give it a shot with our material to see if they could take one last chance trying to heal this wound. You can see they took the material here day 0, sutured it into the wound bed after debriding it and cleaning it up. And within just 1 week, they were able to generate enough tissue using the matrix to actually form over the met head and actually start to close that wound. And eventually with 2 applications, we were able to completely close this wound and actually save this gentleman's foot from amputation. So it's a great example of how this type of technology and how this type of product can really serve as a positive, have a positive impact in patient care. And then lastly, another, one last case I'll show you here. This is actually a case from South Carolina. This is a young woman who had a burn over the back of her neck and her shoulder, which was treated with the material. And we were able to get, full closure of that wound, healing of that burn wound, and also promote that repigmentation as well. So another great application for the technology that we're sort of looking at moving forward. But hopefully overall, this kind of gives you a sense of not only kind of how this material, this technology really provides a new solution in this space, but also how it really can provide some added value for clinicians and patients alike. So with that, before I turn it back over to Pravi and take a few questions, just wanted to sum up a few kind of final thoughts. I think overall, I think we're just really excited about this technology and think there's so much potential for this technology in a variety of different clinical areas. I think it's been a great journey to see this technology come all the way through or come sort of full circle in starting out in the lab and now coming all the way to full commercialization and see it now impacting a number of patients across the country. And personally, while I never envisioned myself going down this entrepreneurial pathway, I found that this path has been really rewarding and simultaneously offers a number of great ways to impact patient care, even though you're not sort of one-on-one with patients every day. But again, I think all of this came at some of the important sort of foundations and lessons that I learned back at my time at CASE. And so without CASE and without the BME program, I'm sure I never would have been able to go down this path. So with that, I want to thank you all for your time and for letting me sort of share my story today. And I'll turn it back over to Pravi to see what questions I can answer about our path or our technology or our business.
Thank you, Dr. MacEwan, for such a great presentation. I think something a lot of people are curious about is like going from like a startup to like such a big company. There's so many struggles you must have encountered along that journey. I was wondering if you could share about like some of the personal struggles, like to like work-life balance, raising a family, like any of those kind of struggles that you kind of faced while founding this company, basically.
Sure. No, it's a great question. You hear a lot about sort of what the startup lifestyle kind of looks like. I think there's a lot of focus on it now, but it really is true that there's a lot of highs and a lot of lows. There were definitely times as we were first starting the company, and we're thinking through sort of the potential risks and benefits of jumping from the university setting into the company full time. And I know that my wife and my family had a lot of questions about what that would look like, and will we be able to support ourselves in going this sort of risky way. And so I think there was a lot of thought around that. But I think as I look back on it, I think this has been such an invaluable experience. We've learned so much about the process of bringing these technologies through to commercialization. And I think no matter what happens sort of moving forward, I think this experience has been such a great one, and it's definitely been worth sort of that initial risk that we took early on. Something else I'll say is there's definitely been a lot of tough hurdles that we've had to overcome, whether it's early in the early days raising capital and making sure we had enough to pay our staff and our team. There were definitely months where we went without pay until we kind of met that next milestone. But then there are also great highs along the way too, in terms of getting these initial FDA indications, or even just sort of seeing the impact of the technology on some of these patients. And so for example, some of the great benefits now are I have patients who call me every week and tell me how they're doing and tell me how their wound is, and it's so great to sort of see that now. But yeah, there's definitely a lot of ups and downs associated with the startup path. But in the end, I think it's a great path that you get to learn a lot. You get to see sort of all the aspects of the business, have your hands directly in sort of the development and implementation of technology. And I think those are some of the really unique aspects of the journey, I think, that you maybe couldn't get in other professional settings.
Thank you. Before we move on to the next question, does anyone else have any questions for Dr. MacEwan? Anything they would like to ask? Yes, Casilda.
Yes, yeah, that's an incredible presentation. I really enjoyed it. Thank you, even though I'm layman in the subject. I'm wondering about the status of the company as an entity, as a financial entity, and where are you going to next?
Sure. So you mentioned that the titles. What I don't think I quite understood your question.
Just where is your company? Are you still raising funds, or are you already preparing to IPO? Or
yeah, it's a great question. So we're sort of looking at a number of different options right now. We have a great sort of set of backers right now. So we're always, I think we have a lot of the great resources we need to continue our growth. But we are sort of looking at different options moving forward and considering a number of the ones that you sort of mentioned. I think there's a lot of potential right now in this space. There's a lot of interest in the regenerative medical market, and so right now we're sort of looking at a few different options along those lines. So at this point we're sort of continuing our growth and looking for other partners as we move forward.
Anyone else have any questions?
Yeah, I have one. Dr. MacEwan, these results are incredibly exciting, and I was just very curious about what challenges do you currently have to make this standard of care. So is this covered by Medicaid and insurances? Is it FDA approved? And what do you need to do to make this available to more people?
Yeah, Jessica, that's a great question. So one of the biggest challenges we had over the past, I'd say, year or so was reimbursement through CMS and Medicare. So because this technology is very new to the wound care space, there were no sort of classifications for this type of technology under CMS's reimbursement. And so we had to work very long and hard with CMS to actually advocate with them and show them the value of the technology and that they should reimburse for this product in Medicare and Medicaid patients. And so luckily, though, we were able to actually work with CMS and get a whole new product class of codes created. And so now we have a path for reimbursement of this this material that we really kind of spearheaded. Now that's a great step forward, but there's still a lot more to go. Something right now we're looking at is how we can get reimbursement in other sites of services, beyond just outpatient care. So that's definitely a big piece that we're continually working on and working on with CMS. The other thing I would just say is, this is still pretty new, and I think a lot of the wound care professionals that we work with are still more used to the human allogenic products or the animal-based products, and so we're constantly working to educate folks and show them the clinical data and continually kind of get them to think about this as another option in their treatment paradigm. And so I think that aspect of education is really important as well, just getting it out and getting it in front of more and more clinicians and show them that this really is a not only clinically effective treatment, but also a very economic treatment, and may potentially have ways for them to save money and save cost in treatment down the road. So I think the education point is really important, and then also the reimbursement is a big thing, and working with CMS moving forward, that's going to be one of our top priorities.
Thank you.
Anyone else have any questions?
I had a question.
Sure.
So Matthew, forgive me if I'm actually, I guess if you covered this, sorry, but were you sort of like practicing as a clinician looking for a solution to a problem, or were you initially involved in the research of this technology?
Yeah, so it's a great question. So no, I was more on the technology side. So I was going through medical school when I sort of stopped and did my PhD in biomedical engineering, and that's when I sort of first started working on this in conjunction with Dr. Shah's lab. And so I was really more involved in the initial technology side. So what was interesting though was I sort of got through my training and was sort of had to make this decision about going down this clinical path or kind of jumping out to help run and form the company. And so that was sort of one of those tough moments that Pravi mentioned. I'd always envisioned going down this path toward clinical medicine and impacting patient care on sort of a one-on-one basis, but I really want to see this technology kind of move forward, and to do that really felt there was a need to have that company as kind of that bridge to get the technology into the clinic. And so that's when I sort of decided to take a leave of absence from the clinical side and really focus more on the technology. So So really my day to day now is really just focused on getting this technology again, out more broadly into the clinical space. And but as part of that, I get to work, sort of hand in hand with a lot of clinicians across the country and hear how they're using it, see what questions they have, and so it's kind of I still sort of stay in that world, but I'm not sort of the primary person sort of using this material day in and day out in my own practice.
Gotcha. So a follow-up to that is, how did you actually evaluate whether there was a pathway forward from research to market, and actually like whether this business would be feasible?
Yeah, that's a great question. So I went through a program at WashU that was offered by the Scandalaris Center, which is the entrepreneurial program here, and it sort of took you through that path of sort of looking at the competitive landscape, doing the IP analysis, looking at how the intellectual property fit against other things that were that are out in the market, and then also looking at doing sort of the market analysis and see sort of what the market opportunity was for this. So we kind of worked through all of that kind of sort of early on. But I will say that, like, I never had sort of the formal business background. I never took any business courses. And so in a lot of ways, I just sort of, I would take 5 minutes and talk with anybody who would give me the time of day, tell them about my crazy idea for using these materials in tissue repair, and in a way kind of piece together all these parts, bit by bit. And I think in general, a lot of that sort of de-risking and evaluation came from that sort of collection of discussions and interviews and sort of I guess sort of collaborative work that we did here in the local kind of St. Louis entrepreneurial ecosystem. But definitely tried to early on kind of lay out the opportunity, understand the risks, the potential, and then sort of map out what that path to market looked like. And really, I think some of those early plans and evaluations that we did, a lot of that really held true. I mean, there were definitely some bumps in the road, that came up that sort of threw us off course. But along those lines, though, I think generally speaking, a lot of that early work really gave us the right direction and path that we ended up following.
Thank you.
Thanks for the question.
For the great talk. Just to have one last follow-up question. Do you have any advice that you can give for any people who are planning to have a startup right now, or building a company, or any new entrepreneurs out there?
Sure, it's a great question. I think that the best advice that I could give is that I think first of all, I know a lot of people sort of worry about when's the right time to start, when's the right time to begin something, and is it worth the risk, right? And I'd say that the best thing you can do right now is really just get moving, get started, and really take that leap, because it's a very rewarding pathway, whether your startup or your technology works or not. It's a amazingly valuable experience. And gives you a lot of insight and access to a number of different areas. So it's a great sort of a learning experience there. But the first step is really just to dive into it. And so I would recommend to anybody, it's really worth going down that path. And additionally, even though it's sort of maybe a less traditional route than some, I would definitely sort of keep it on your radar, and also be just open to other possibilities, because again, this wasn't sort of the direction I thought I'd be going in, but really turned out to be a very rewarding experience, and one that I'd recommend to anybody.
Thank you. And I think now Doug will say something.
Thank you so much, everyone, for joining us today. Thank you, Matthew, and thank you, Pravi, for taking the time to lead this session. We greatly appreciate the opportunity to learn about your career journey and how you've intersected with entrepreneurship along the way. Next week, we have Ross Pillary, who's the former chairman and CEO of BP America, who's going to join us at 12 o'clock. Please feel free to sign up. And as always, have a great rest of your week, and we hope to see you again.